Good morning, children. Today's topic is culturing method for the heterogeneous host which is having gene of interest to produce the protein. So culture is done in two ways. One is the batch culture and another one is the continuous culture. In the batch culture, as you have already studied here, it will be similar like that. This is the pipe which is providing sterile air, so no contamination of any other bacteria is there, only the bacteria which we require for the production of protein having the gene of interest is there, and these are the mixtures which will keep on mixing and providing the nutrients to the bacteria. Now in the batch culture, as in the beginning there is some bacteria, so growth will be slow. Let us first understand with the help of graph. This is the bacterial growth and this one is the time. So bacteria will consume the nutrients and will grow slowly in the beginning as very less number of bacteria is there. So it is known as lag phase. Later on the number of bacteria will increase and as by the mixing the food is being provided to bacteria, so the growth will be fast and it is known as log phase. Once the nutrients are consumed by the bacteria, all nutrients finishes, and also as bacteria is growing, so waste will also be generated by the growing bacteria. So the waste generated by the bacteria and also the nutrients being consumed, then the bacterial growth, because of that the bacterial growth will stop. It will not grow further. Now in the culture method it is different from that of the batch method. It is more efficient. What happens here? Here in the vessel, from one side, continuously the nutrients, the nutrient medium is being added in this flask, and continuously the waste is being removed from the other side. Here also the aeration pipe and all will be there, the sterile air will be passed, the mixers will be there and all is there. So some bacteria is here. Let us understand this also with the help of graph. This is time and this is bacterial growth. So as there is some bacteria, so growth in the beginning will be slow. It is known as lag phase. Number of bacteria will increase. Also the sufficient amount of food is being added continuously from the other side. So they will have the sufficient amount of nutrients. The growth number will increase. So the growth will be fast, and that is known as log phase. Now the difference here is that the growth will not stop. Here the growth stops because of the consumption of all the nutrients and accumulation of the waste by the bacteria. Here it will not stop. The growth will be exponential, and as the growth of the bacteria will be more, production of the protein will also be there. From this side, removal of the waste product is done. Three things are being removed: removal of the protein product which is being formed, removal of the waste generated by the bacteria, because accumulation of waste will kill bacteria and bacteria will start dying, and also some amount of bacteria, few bacteria are being removed from this side. Why? If the concentration of the bacteria increases, the shortage of food may happen, though the nutrients are being added, but bacteria grows very fast. So from this side some bacteria will be removed. If some bacteria is removed, removal of waste is also done, so bacteria will not die, and the protein product formed by the bacteria is also removed. So exponential growth of bacteria will be there and production of protein will be continuous. So therefore continuous culture is more efficient. Now what happens here? Removal of the waste generated by the bacteria and the protein product produced by these bacteria, and also some bacteria needs to be removed, as you know, otherwise the shortage of nutrient will be there. So all are removed, are mixed together. They need to be separated. Only thing is that they need to be separated here. Thank you, children.